Now I will present Professor Lehn. So Professor Lehn was born in 1939 in Rosheim, in the region of Alsace, in France. So at the beginning of his education, he was hesitating in coursing either the high school studies in experimental science or in philosophy, actually. So he did philosophy, and in the same year he also did experimental science. So he did both at the same year, actually. He finally decided to dedicate his life to chemistry and obtained a PhD in 1963 at the CNRS in Strasbourg, moving then for a postdoc at Harvard University afterwards. He was appointed as assistant professor in 1966 at the University of Strasbourg and started his independent research in NMR studies. From here, he was interested at the very beginning, in cation permeation through membranes, and subsequently in different research lines, he coined the term supramolecular chemistry for the first time. So he became full professor in 1970, and he has mainly shared his research between two laboratories, one in the University Louis Pasteur in Strasbourg, and the other one in the Collège de France in Paris. He has published more than 800 scientific articles, and most of the current recognized researchers in supramolecular chemistry are actually come from his lab. The number of awards, decorations, honoris causa is huge, is enormous. Of course, we highlight the Nobel Prize in Chemistry in 1987. So, as you will realize in a moment, concepts such as self-organizing systems by design or by selection, or constitutional dynamic chemistry, adaptive chemistry, and molecular recognition have been concepts pioneered by Professor Lehn. So all chemists, but also biologists and science in general, should thank you for your 50 years of career. And we are very happy to have you here in Girona, and we're looking forward for your talk.
Okay. It's very nice to be back in Girona. I've been here twice before, but never at the university. So, Xavi, thank you very much for inviting me. I'm very impressed for what's going on here. Small place, but small is beautiful, it looks. I think, is a representative of the mayor still here? No, because I think Girona is a small city compared to Barcelona. It's like Strasbourg compared to Paris. But you try harder, Anna. You try harder, and you may get better. It's a competition in science, and you have to be better than Barcelona, okay? Who is undergraduate here? Lift a hand. Undergraduates. Graduates? So most of, yeah, okay, the other ones I know, these ones in front here. Because I have changed my lecture somewhat. I was originally more hardcore chemistry. Now we start a bit broader and tell you also why we are chemists. Because we need at some stage some justification why we do what we are doing in our life. And I would like to convince you that chemistry is a very important part of it. So I changed my title from perspective in chemistry to something which will be seen is very close. So half of it will be very general, and half of it will be, again, something more for the advanced people. So it has to do, I have to go up there because this thing. Start. Ah, oui, okay, that helps. Yeah. So, it will be much more general towards complex matter. Now, what does that mean? First of all, the question, has chemistry something to do with that? And we have also to define what is complex matter. And the answer is chemistry, of course, has something to do with that. And for that, we will start very early. The beginning of our universe. 13.7 billion years ago, there was a Big Bang. And after that, energy was generated, particles formed. The universe cooled down very quickly. But at the beginning, there was no chemistry. It was only physics. It's the age of physics. Particles formed. They aggregated to make atoms. when atoms were able, because of the temperature which was lower, to get together to make molecules, then chemistry started around here. The start of the age of chemistry. Then the universe continued to change, and molecules got together to make aggregates. These aggregates at some stage defined primitive membranes. Out of these membranes, some property came up which was progressively going toward what we call life, and then biology started about there at the planet, on the planet Earth, the age of biology. But on our planet, it didn't stop there, as we know, because something else appeared, which is thinking. So before I continue, this is, who knows what is this? Very famous statue. Le Penseur, the Thinker by Rodin, Auguste Rodin. That's one of my preferred ones, because it's really mankind thinking about what mankind is doing here. It also points out to one thing. This is the way we look now. And some people want to stop the evolution of the universe at that point. That is impossible. In 1,000 years, we will not be the same, I am convinced. We are not the end of evolution. We are only a point in space and in time as we are sitting here. So some people get afraid and say, stop it. You cannot stop it. Okay? You have to realize that. So yes, you cannot stop it. The best is to go with it. Because if you go with it, you can control it. If you want to stop it, you can't do anything anymore. Furthermore, I'm personally convinced that there's life on many planets in our universe. People have now found thousands of exoplanets in other galaxies. A number of them have the conditions the Earth has. So, the other people somewhere up there may be giving a talk right now. So, this is the way to complex matter. Starting with the very simple constructions, a very simple part of the universe, and coming to a thinking organism. Then a big question arises, which I consider perhaps as the biggest question in science. Of course, we know that there are very fundamental explanations of the universe, like Einstein's general relativity or quantum physics. But the most fundamental question, I think, is this one. How does matter become complex? How can you go from an elementary particle to a thinking organism? Because after all, because this is possible, that's the reason why Einstein existed and why Planck and the others existed. So the more basic question is, how is it possible for our universe to generate something which is called a thinking organism? After that, you can do the rest, of course, but first you have to have a brain or some kind of a device which can do that. So, in this investigation of this question of how does matter become complex, physics deals with the general laws of the universe, the ones which regulate everything, the ones on which we all depend, that's the basis. Biology tries to understand the rules of life on our planet, our own life. And chemistry can build a bridge between the two. In other words, the job is to try to understand how from very general laws, which can do anything compatible with the laws, something can emerge like the organism we have on the planet Earth. And what's the process? How is this? How has it happened? How can we do about? How can we understand it? The answer to that question is by self-organization. Now that means everything and nothing. Self-organization, we understand, means that it happened by itself on the basis of the structure of our universe. That the laws which drive the universe lead to self-organization. But that doesn't mean we understand what's going on. So now we have to try to understand what this is, this self-organization. What are these properties? What has matter done in our universe to be able to be self-organized? So chemistry is in sciences. Mankind has created what is called science to understand what we are doing, understand us, understand the universe. And within these sciences, as we cannot do everything, I guess most of us here would like to be mathematicians, physicists, biologists, chemists, and all that, but you cannot. So we are chemists, and our job as chemists is to try to understand the structure and the transformation of matter. That's one definition of chemistry. I think it's a correct one. But there's another property which I come by in a moment, which you will see is very important also. Now, these elements which form the universe were discovered progressively. People didn't understand exactly what they were in the 19th century until came up the idea, and Lehn was the first one to understand that there was an order in the elements. And he published in 1869 a very famous paper. I consider it's one of the most important papers in science ever written, because he said that you can organize, more or less I summarize, you can organize elements. In that case it was according to their atomic weight and their properties, but anyway an organization came out which led to in fact that's the original paper here. I think you should read it because I think it's a fundamental paper where he begins to organize the elements. For instance, here is you see here, beryllium, magnesium, there is calcium somewhere, and here you have boron, aluminum, and so on. But Mendeleev went even further. At the time when he proposed that table, in his scheme there were holes, something lacking, question marks. And he said we will find them someday. Of course, they were found. Nowadays, that's the way it looks. Both of you, I hope, knows the periodic table. But I want to insist on one thing. This is the most important table science has generated. Why? Look at this. It tells you everything in the universe is made of these elements. In our universe, there is nothing else as a building block to make up matter in our universe. It's very interesting. You have to think deeply about the fact that you are in front of the table which represents everything from the planet Earth to the end of the universe, billions of light years away. Now, why is it so? Because 1, 2, 3, 4, 5. There's nothing between 1 and 2 and 3 and 4. What is 1, 2, 3, 4, 5? 1 proton, 2 protons, 3 protons, 4 protons, and around it 1 electron, 2 electrons. The periodic table is just counting. And there's nothing between 1 and 2. So the periodic table is finished, it's full, and it contains everything. And science has produced that. Now, that's the playground of chemistry. As chemists, we can use those things and play with them. And of course, in the development of complex matter, evolution of the universe has played with this, depending on the conditions. So initially, chemistry was trying to take atoms and make molecules. Take bricks, make houses. And this is molecular chemistry. This molecular chemistry uses strong bonds between atoms to build up complex structures called molecules. And these strong bonds were the way to build up these houses, these architectures, and it is a sort of an art and science of organizing molecular matter. How can you organize atoms to make molecules of very various types of complexities, natural ones, but also ones which are made in a laboratory. And chemists have made thousands of molecules which do not exist in nature, just because it's possible, and because it follows the basic laws of physics and the imagination of the chemist and the properties we want to have. Now, just 2 milestones in this progress of having more and more complex structures generated. On one hand, urea was synthesized by Friedrich Wöhler in Germany in 1828. That was a big achievement at that time. He used an inorganic compound, ammonium cyanate, to make out of it urea. He made the synthesis, ammonium cyanate gives urea. Second, very important step, you mentioned philosophy, very important step on our look at the universe and what happens, people at that time were thinking that you cannot make anything contained in a living organism without the help of a magic force called the vital force. Wöhler showed that you could go from a non-living, from an ammonium cyanide, which is not contained in any living organism, to urea, which is contained in urine. And therefore, molecules are molecules. You can transform them into one another. There is no vital force. It was very important at the time. 150 years later, in the 70s of last century, this very complex molecule, vitamin B12, was synthesized, produced from the building blocks by combined efforts of Robert Woodward and Albert Eschenmoser. Robert Woodward at Harvard, Albert Eschenmoser at ETH in Zurich. They had about 120 collaborators. It was a very long synthesis, 82 steps or something like that. This is my part of it. I was postdoc with Woodward at that time. And yeah, okay, it's in the final structure, it's my part. I want also to indicate by that if you are a scientist, you make a contribution to knowledge. And what you bring may be a small stone or a big stone, but it's your stone. So I would like to insist that participating in this enormous project, which is building up knowledge, is like bringing a stone to either a synthesis or a huge building or some kind of nice construction. So this is molecular chemistry. How to make more and more complex molecules. And molecular chemistry has, of course, continued since 1970s and has become much more powerful. Many new reactions, many new materials, many new molecules, many new properties and all that. And it's very strong science and adult science, and it's continuing. But you may ask a question, is there something else we should think about? This is a cancer cell. These are two killer cells. The killer cells have a job. They have to find the cells which have gone wrong and destroy them. How do the killer cells know that the other one is a cancer cell? What tells them? If they make a mistake, you have a problem. Either they don't destroy the cancer cell, or they destroy healthy cells. No good. So how do they know? These blue dots is the HIV virus, which are now attaching to a white blood cell. When the HIV virus reaches the blood cell, it can infect. How does the virus, let's say, know that it has reached the target? So something must happen between these objects, the killer cells, the cancer cell, the virus, and the white blood cell. And this something can only be as they get together, as they touch. Now when they touch, that means that the molecules making up the surface, defining these objects, these membranes, they contain molecules which get in contact. And it's this contact which is now this new property which makes molecules recognize, feel the other one. And this is done, the field which we have defined as supramolecular chemistry, where the important thing is the step beyond the molecule, where the question asked is what happens when molecules get together. And this also raised the other question that, like chemists since, let's say, 1800 something, have learned how to manipulate covalent bonds to build molecules. The question then comes, how can you manipulate non-covalent interactions, intermolecular interactions, to build up larger structures? What happens? How can you take control over that? And three main features have been studied in this first step. They are, how do molecules recognize each other? How do they act on each other? And how can they carry each other through a barrier, like a membrane, an artificial one or a natural one? Recognition is the most basic feature. How do molecules recognize each other? And this process is extremely important because there is no biological process which does not start with a recognition. This is fundamental. If not, everything goes wrong. So the interactions which one has to manipulate, in molecular chemistry, you have CC bonds, CO bonds, CN bonds, and all that. Here you have electrostatic interaction, hydrogen bonding, donor acceptor, and so on. These are just convenient ways of dividing up the interactions. A physicist would tell you all that's the same. That's electron distribution, these interactions, that's the Schrödinger equation. Yes, but you can't do much with the Schrödinger equation when you have big molecules like that. So it's convenient to do this and to think about these different types of interactions, which indeed are sufficiently different to be able to be handled differently. I don't want to talk about medium effects. Now, molecular recognition is the most important. When molecules get together, how do they recognize? How is there a selectivity in this interaction? First of all, you need interaction. You need that they talk to each other. If they ignore each other, nothing happens. So there has to be an interaction between them. That's number one. But very importantly, there's a second property, information. You cannot have recognition without information. In other words, all molecular processes where molecules get together is an informed process, is an information process. And this can most simply be expressed as a double complementarity between the geometry of the object and their interactions. Geometry is simple to understand. They have to fit together. And second, they have to have also they have to fit together geometrically. But second, they have also to fit together in terms of the interactions. A plus attracts a minus. Two dipoles attract when they're like that. They repel when they're like that, and so on. The simplest explanation, a very strong image, was already given by Emil Fischer in 1894. For all those here who are biologists, I guess they know it. For those who are not, he published in 1894 a very famous paper where he said for an enzyme to act on its substrate, they have to fit together like Schloss and Schlüssel, like a lock and the key. This is, of course, we now know that the lock and the key are adaptable and so on, but basically, locks and keys, it works. Knowing that it's not absolute, but it's the basic image. And this is this basic principle, the basic image. And here is Emil. He got his PhD in Strasbourg in 1874, when we were part at that time of an empire across the Rhine. At that time, we belonged to Germany. We were not French for 50 years, but we had fantastic scientists. And Emil got his PhD there. And 20 years later, he wrote that famous paper. He's one of the greatest organic chemists who ever existed. Now, recognition. Let's take an example, which I hope all of you know, but it's good to mention it. We as living organisms are all dependent on our genome. Our genome is what makes us different from a tomato, or from a bacterium, or from an elephant. And what is it? It's a program. It's a program which is written with just 4 letters. Trivial. The 4 letters are 4 chemical groups, which any organic chemist would say, oh, that is very simple chemistry. Indeed. Adenine, guanine, thymine, cytosine, very simple heterocyclic groups. And these 4 letters and their sequence on a long strand, which is wrapping around each other to make this famous double helix, that defines all living organisms. So that's a minimal approach. Why just 4? Why not 6? Why not 10? Because 4 is enough. And often evolution stops when it's enough. It doesn't explore further. And now we can do it. And there are people trying to add letters now to this alphabet. Now, how do you read this information? You have to find a reading scheme, which is also trivially simple. Pairing. If you have 4, you can make 2 pairs. And if the 2 pairs are different, you have a reading mechanism, a binary reading mechanism. 2 hydrogen bond interactions, represented by the dots, or 3. It's a binary system. In a computer, it's 0, 1, 0, 1. It's 2, 3, 2, 3. Same thing. So the storage and the processing of genetic information is chemically, trivially simple. But it's enough. And we know that it works. So chemistry, one can claim, is an information science also. There's molecular storage and supramolecular processing of information. So I can say that chemistry is a science of informed matter. Of course, this information, any molecule contains information in its shape, in the disposition of the atoms, and the processing of this can occur by the contact between the molecules. And supramolecular chemistry has to deal with this, how to store information and how to then process it. Now let's just give an example. We play with locks and keys. We use the good old Emil Fischer image. These are keys, that's a lock. Obviously that key fits in. So when we started this work, which is sort of some time ago, about 50 years ago now, my lab has its 50th anniversary next year. So the idea was, what is the simplest object to recognize? In three-dimensional space, what is it? A sphere. The sphere is the simplest thing. So what are the spheres in the periodic table? The spheres are, for instance, alkali cations. Lithium, sodium, potassium, rubidium, cesium. A very nice sequence of spheres of increasing size and just one charge. So everything is constant except the size. So can we recognize a sphere in a collection of spheres? There was another reason why this was very interesting. Two of those, sodium and potassium, which are next to one another, are very important in biology. At the beginning of that work, I was interested in neuroscience. And one approach to neuroscience was to understand how the nerve influx propagates. How does it propagate? When I do this, there's a signal going through. It propagates by changing sodium-potassium concentration across a membrane. That's the action potential. And there must be molecules in the nerve membrane which are able to make the difference between sodium and potassium. Otherwise, there's a short circuit. So there is something which makes them able to select sodium content and to control the efflux and influx of these ions. So that was also a biological reason for being interested in sodium and potassium and in this range, and therefore in spherical molecular recognition. So to make a long story short, it's a very long story, because 50 years old now, and this is the first molecule we made, a cage, which, like the lock, has a cavity. This is the molecular model. It's nicer to see on the molecular model. Has a cavity just right for fitting in potassium. This is a lock and a key. That's a lock. This is the key, and that is the combination of a lock and the key. And we call those cryptands and cryptates because they're crypt, they're cavities. Here are crystal structures. The crystal structure with the rubidium was made, but potassium, same, and so on. These are the exact features. And then, of course, you want to adapt your lock and you synthesize these things. That's another power of chemistry. If you ask a question, you can get an answer by making the object. The idea there was, maybe these cavities are good, and we can control them. Don't you make them? This is a fantastic possibility. The possibility to make what you can test your hypothesis. That's like physicists, when they discovered the Higgs boson, that was to test the standard model. Without the Higgs boson, they would have a problem. And so I think science can do that, testing your theory, testing your hypothesis by doing the right experiment. Here we have one small cavity binding lithium, the one sodium, the one potassium. Now this has developed a lot, just one, I haven't time, there's hundreds of papers, thousands of papers have been published now in this field where people have made artificial molecules which are able to recognize their keys, so to say. And this is just another one to show just an example. That's ammonium ion, that's now a tetrahedron. And then you can synthesize a molecule which has 4 binding sites and located in a tetrahedral fashion. That's the molecular model of that. We have a crystal structure also. And you see this is a very nice, in fact, it's one of the nicest fits we have ever had, because the ammonium sitting in here has just the right shape, tetrahedral, the right size, and the right interactions to just get in. Now, I could talk for a month on these things, and there are now volumes, 12 volumes on supramolecular chemistry, so I don't think we will spend time on all 12 volumes. But what about some applications? We heard a very nice talks earlier on use in biology. We heard a talk on also nanoscience and nanotechnology. And I think, obviously, there are consequences to this type way to understand, try to understand what happens in interactions. And what we heard about, everything was interactions also. So what about some application in life science? The most obvious one, which I haven't time to get much into it, but there was a little bit more later, is of course drug discovery. Because after all, what is a drug? A pharmaceutical bioactive molecule is a key for a biological lock. You want to make a key which goes somewhere as selectively as possible, isn't it? Just to reach the right point and to do something at that position and not going somewhere anywhere else. That's idealistic, but that is the basic use, so to say, of understanding molecular recognition to design bioactive molecules. We know that many of our drugs have come by trial and error. And from natural substances, you extract them, you try them, and well, some work, some don't. But you can also try to understand, and that involves molecular recognition. Second one, I want to give you one example of the use of what we have been doing, and a third one. This example has to do, again, with something which sort of is almost unpredictable. We were interested at some stage in photophysics, in trying to understand a process which can occur in molecular cages. So we had made this cage, which is a cryptate, but of another type, and put europium ions inside. Europium has a red fluorescence, but it's a very, very weak one. So when you put it in the cage, it's much, much stronger because energy is absorbed by the outside and transferred to the europium, which then emits light. And this can be used as a label for monoclonal antibodies, which then can be used to do an immunoanalysis. This system was developed by Girona and his company, and this machine called Cryptor is now sold by another company, Cézanne, in Nîmes, south of France, not far from here. This used in many hospitals now. So that was one of those things when he started cryptates. Of course, we didn't think about immunoanalysis. First of all, because monoclonal antibodies didn't even exist at that time. So this is one other example. It's another one coming up later where fields develop, and at some stage you see connections and progress can be made because you bring things together. Another case of that is this one. We have been studying transport processes, artificial transport, to try to understand how also cell transport occurs in many cases. And then we dropped that in end of the 80s because it's done a lot. And then came up a little bit later the fact that it would be very interesting to introduce pieces of DNA, genes, into cells, both for biotechnology and for gene therapy. What is the problem? The problem is that the DNA is a very highly charged negative molecule. It doesn't go through the membrane. So what you want is you want to hide the charge, to envelope the DNA, so as to have it go into the cell. So you want to make a synthetic vector which will go around, hide the charges, help it to go in, and then, of course, I don't want to get into that. It's very complicated processes. You have to go to the nucleus, then you make the RNA, then you make the protein. But like one often can do, you'd use a reporter gene which gives a protein which you can easily detect, like beta-galactosidase, which allows you to color cells in blue, or luciferase, then the cells can emit light. They're fluorescent. And this is now very important for biotechnology and also for gene therapy. There's, in biotechnology, many studies are done and many companies are interested in that, of course. So we developed reagents for that, and one could see that indeed with rather simple molecular vectors. And many have been made around the world by companies which now are sold around, where you can facilitate the expression of this gene and get the resulting protein. I don't want to get into details because, again, it's a field in itself now. It's very important also for gene therapy, especially for some genetic diseases like cystic fibrosis, where one can indeed show that you can modify you can do gene transfer in the airways of mice. There's one of my brothers, who's a cell biology professor, did that, and you can then check that you can transfect your lung epithelial cells. Now, once you understand a bit more about molecular recognition, comes another step. Can we use these molecular recognition features in order to make systems which will self-organize? Systems which will generate a complex architecture in a spontaneous way, but controlled. You want to control it. You want it to be spontaneous, that means to happen by itself, self-organize, but you want to control it. This self-organization can be, okay, I don't want to get into equilibrium, non-equilibrium, there's already another step which is a complicated one. So let's just stick with thermodynamically controlled self-organization. An example, a very simple one, is how a virus builds up. I took an example which was the first virus sort of understood, how tobacco mosaic virus builds up. Tobacco mosaic virus is an aggregate of molecules, 2,130 proteins go together. They have this sort of conical shape. They touch surface to surface. They interact surface to surface. So they go from a piece of cake like that, a piece of pie, going together and generating a disk, a log washer, and then a helical tower. When you look at this, you say, oh, this is magic. It's not magic at all. Our today's chemistry explains everything that is happening here. Because the interaction to the surface, in that case, it is self-complementary. One side of the molecule is complementary to the other side, so they can get together. They are shaped in such a way, when you have a piece of cake which has this place, if you put them together in this way, it has automatically to go around. Huh? And so that's the way it builds up. So it is not magic, it's just the basis of structural chemistry, organic chemistry, physical chemistry, and it happens. And thermodynamics, of course. This again looks like a program. These bricks have information. This is being read by the contact, and you generate the virus particle at the end. So the information is in the components. The processing of that information is supramolecular by interaction between the components. You can even define an interaction pattern, which is a recognition algorithm. I come back to that in a moment. So you input information, recognition occurs, the substance grows, at the end you have the final particle. So it's a sort of a program system. And again, I want to insist that this is functioning like a program. And this is chemistry. Now, let me give an example which deals, I chose one of them, which is the sort of the simplest one, where we use, and it's also totally non-biological, which also indicates how one could do things which have nothing to do with biology. Just to show that the world of molecules is a much more wide open world than the ones which are realized in living organisms. And so let's see what we can do. We will use molecules able to bind metal ions, and the metal ions will serve as cement, connectors to bring them together. So the metal ions will process this structural information which is in the molecule through what you may call the code. An inorganic chemist would call it a coordination geometry. But the coordination geometry is a reading algorithm. You can talk it. It's another way of talking about the same thing. But I think it's sometimes important to change your language because you understand things differently. Coordination geometry is also a reading mechanism, and we'll see why. First of all, I need that for something else, but the first thing we did was to try to make double helices. DNA, that's emblematic, also a mystical object. But can you make double helices which has nothing to do with DNA? And with units which are totally different from DNA. Of course, they don't have the same function. That is quite clear. Yeah, you can take a strand of 3 subunits. That is like a trinucleotide. And you can use metal ions which binds to these subunits. And when you use the right subunits and the right metal ions, you get a double helix. This double helix, that's a crystal structure. It's not an image. It's a real shape of the molecule. You can also make a little bit different strand, slight change. You treat it now with a molecule which requires 6 coordination. I should have said this copper one requires 4 coordination. So N, that's 4. So it wraps around in this way. nickel or cobalt or iron wants 6 coordinations. So you need 3 times 2. 3 strands, 1, 2, 3, 4, 5, 6, and you get a triple helix. So you can make double helices, you can make triple helices, you can play around, and you can develop a whole sort of a field, which we call the field of helicates. We call those things helicates, and you can play around with the information present in these basic units. Now, I go now to another aspect of it, because the first one was sort of had to do with some aspects of structural biology. Now we can look at something which is closer to nanoscience and nanotechnology. When you look at these things here, this looks like a small device, a circuit or something, where you have perpendicularly oriented wires, and at the intersection, they something, like a semiconductor piece or something. You put something there, which links them together. So you need to make objects, molecules, which have here slots into which you can insert a metal ion. If you build that correctly, and if you use the correct metal ion, you will get this organization. And one way to do it is this one. You make molecules which have 1, 2, 3 binding sites within a given subunit. That's one subunit. That's the other subunit. Now, here I can explain a little bit this question of algorithm and so on. I could do it earlier, but that's a good example here. Why do these things orient perpendicular? Simple geometry. The metal ions you use are hexacoordinated. That means they form an octahedron. What is an octahedron? 2 times 3 points in 2 perpendicular planes. 3, 3, that's an octahedron. So, if you have 3 and 3, if you have 3 as a subunit, if that binds to one metal ion, the other one has to bind perpendicular. So it's obvious. You have to get this, and you get it. This is an example which just shows that you have to handle the structure of the ligand, the structure, the binding features of what brings it together, and then you get automatically these objects. These objects are then multi-ligand, multi-nuclear. There are many ligands, there are many metal ions. There can be different metal ions. I haven't time to get into all that. And let's just have a look at the simplest one, which is the one which is the 2 by 2 grid, which you can form by using these objects, which I just showed. They assemble with various metal ions, cobalt, zinc, and so on and so on. And that's a crystal structure showing that indeed it forms. This is bulk. Now, how do you talk? Can you talk to these objects? Can you try to titillate them to see what is going on? You can deposit them on graphite. I mean, deposit them on graphite, they form arrays. Let's not get into that. If you look at higher resolution, you see 4 dots here. And these 4 dots correspond to the 4 cobalt ions in this grid, which has 4 cobalts. It's a cobalt 4 grid. In addition, when the tip of your STM machine goes over the structure, the current increases exactly when you are above the cobalt. So it's a way to talk to the cobalt sites there. And this shows that indeed this organization brings up a structure which then can be addressed by, in this case, by touching it with an STM microscope. The largest one we made is this one, which just illustrates that you can do rather complicated things. This has 4 subunits. 4 times 4 is 16. You use lead, and if you mix this with that, you get automatically this structure, which has 8 ligands in 2 groups of 4 perpendicular, and 16 leads at the intersections. 4 of these leads are identical, they're in the corners. 8 are identical in the edges, and 4 are identical in the middle. Lead has a nuclear spin, 1/2. You can do NMR on it. What you see, you should see 1 to 1. You see 1 to 1, 1 to 1. I see the quality of that spectrum is very high because it's not a recent picture, but I wanted to show you that you can indeed address the corners, address the edges, and address the center. Just one other example, you can assemble very complicated large cages. This unit here has 1, 2, 3, 4 subunits. This is linear, this is flat. This has centers, binding sites here. You treat it with copper 1, which wants tetrahedral coordination. And automatically you get this cage, which has 3 cavities, like a skyscraper with floors, and you have apartments at each floor. And this forms just in one step. I mean, I should say in one operation, because in the assembly there are many steps which occur kinetically. But the thermodynamic product is this product here, and here is the crystal structure, where you now see that there are 3 linear molecules, 4 planar ones, 12 midlines. These 19 things just get together and form this entity. I could have given many more examples, and this chemistry of using the coordination properties of metal ions together with the correct type of ligand entities has been very much developed, and structures have been made which 10, 12 years ago people said you are crazy, man. You cannot get it. And you can get it very simply. In some cases, even there's now undergraduate students doing it in the lab. So just a little note about nanoscience and nanotechnology. We know right now that by these very powerful methods of fabrication, of manipulation, nanomanipulation, we can make these very powerful devices, which are our computers. And this, of course, works very well. We know how powerful that is. But we can also think about the fact that maybe there's another approach, which is not to have to make it, but let the object make itself. In other words, to try to understand enough about the pieces, to let them go together, but then that's not enough. The first step has to prepare a second step, which goes to a third step. So there's still a lot to do until we can lead from the beginning to the end device by successive and sort of conditional, because a second step cannot come before the first one, the third before the second, and so on and so on. And this would be maybe the future, a way to go from having to make things to let them make themselves. That would be, of course, the ultimate fabrication. Maybe I can convince you by saying that our brain, which is the best computer for the moment still, is self-organized. You don't make it. Well, it's self-organized. And this complex object is working by itself. So, we'll see. We can do lots of things. Now, this was some examples of self-organization by design. You design the objects which make double helices, triple helices, grids, cylinders, and many other things. You store information, you program the system, and you get out the final product. At least you hope that you understand enough to be able to do that. But there's another approach that you can consider, which is self-organization with selection. Meaning by that maybe the process we are looking at can also select the pieces it needs to build itself up from a high diversity of pieces. So you need a diversity in sets, but you need also dynamics. That means you need to have the possibility to explore. You don't want a system where it gets together in collision and sticks together and doesn't break up again, because you want to explore the thermodynamic, the hypersurface between energy and structure, to be able to find the best spot, thermodynamically the best spot. So you need dynamics. This gives the possibility of adaptation, because a system which can assemble, deassemble, reassemble, when you do something to it, maybe it will change and adapt to what you do to it. And this is a road to adaptive chemistry. We were brought to this idea in the following way. You may remember that this is a strand which with copper 1 gave a double helix. Now, usually when you do chemistry, you want to have pure compounds, which is perfectly justified. But you can also ask the question, these pure compounds, do they have a property which makes them much more powerful than just being pure, which is, okay, that's one characteristic. Then you can mix these together, which is a terrible thing to do when you have spent a lot of time getting pure compounds. Then you mix them and have now, this is the strand which is nickel, give a triple helix. Now you can ask a question, is this mixture still able to generate the correct double helix and the correct triple helix? And indeed, it's the case. So these entities, when they are mixed, they give the same objects at the end, which means that they are able to self-recognize, to generate the correct entity at the end, which is a sort of a dynamic set where everything exchanges and which allows the system to evolve towards a thermodynamically most stable state. The other case which brought us into this step was the following. I didn't mention that we also tried, in addition to make double and triple helices, to make circular double helices, like plasmid DNA, make circles with double helices. And it happened, to make this story really very short, that this strand with iron X2 salt gives a pentagonal structure with 5 iron centers. And in the cavity of this thing, there was a chloride ion. Now we have crystal structure, all that, so everything is correct. But the important thing here is that we get a pentagon with a chloride. And the chloride is very strongly bound. So you can ask a question. This is a brick, this is a cement. Why do you make a pentagon? You could have made a hexagon. When you buy a truckload of bricks, you can make any kind of house. Huh? A big house, a small house, a square house, a round, maybe not a round one, but all types of houses. So why does it give this? So one hypothesis is that maybe the chloride is bound very strongly. So the thermodynamic outcome is the chloride tells the system, I want a pentagon because I can bind very strongly. That means the delta G and the stability is much better. So you do what chemists can do, you replace the chloride by something else. You do the sulfate, you get a hexagon. I didn't put it in because we don't have the crystal structure, but we know it's a hexagon. If you add chloride to that, you go back to the pentagon. If you remove the chloride by precipitating it with silver or so on, you go to the hexagon. So, what is it? It means that you have a library, a dynamic set of, we call that circular helicates, where the initial brick plus the cement can go for a square, a pentagon, a hexagon, and in principle, whatever. But the important thing here is to realize that, in fact, we have this dynamic set which can produce these assemblies depending on what is present. That's obviously an adaptation to the conditions. So this led to the recognition of a property which has always been present in supramolecular chemistry, but which was not really implemented, not used. And this often happens. You develop a field, you take for granted some properties, but you don't realize that there's something behind that, and you have to look at it. And this brought up the question that supramolecular chemistry is a dynamic chemistry. And it's a dynamic chemistry not by kinetics. It's a dynamic chemistry into constitution of the object. The object itself, its constitution can be exchanged. It's not just dynamics, kinetics. It's not just motions. It is a change in the intimate structure of your object. And of course, because there is exchange, the cooperation of pieces, you can change the constitution. This is a dynamic, non-covalent chemistry, which is, so to say, it's normal, it's intrinsic. But then you can have another step. You can again ask a question which at the beginning looks like something you do not want to do. Can we make molecules which break apart? In other words, can we make molecular chemistry dynamic? So now at the beginning, that's something terrible to do, because if you have a mixture, if you have a molecule which has a tendency to disintegrate, to dissociate, you put it in a fridge to cool it down, to prevent it from doing that. But if you let it do it, maybe new properties appear, which you don't have in static molecules. Now, how can you do that? You can try to introduce, in order to import dynamics into molecules, you can try to introduce reversible covalent bonds. Bonds which are still covalent, molecular bonds, but which can open and close, and there are a number of reactions which can do that. We come to that in a moment. So that means then that this is dynamic covalent chemistry. And then we combined the two and put it under just one heading, which is a dynamic constitutional chemistry. It's a bit a complicated word, but what it means, you have understood it, I guess, is that it's the constitution of the chemical object which is dynamic. It can fall apart, it can reassemble, it can exchange its groups. It gets really something sort of plastic, flexible, adjustable. Now, what can we do with it? Just a few examples to convince you that there is a lot behind it. First of all, of course, this constitutional dynamic chemistry generates diversity. You mix it and you get a horrible mess. And in all the times, you put it in a sink because you cannot study it. Nowadays, you have mass spectrometry, you have NMR spectrometry, you have crystal structures, you have all these methods. Now you can analyze it. And now the picture changes. So that's also an example where the progress of technologies to investigate makes possible to tackle problems which otherwise you could not. That is why there are a number of Nobel Prizes have been given for X-ray structure determination, for NMR, for mass spec. And that is very important because the tools are what allows us to go further. I could again do the Higgs boson. Without the Large Hadron Collider in Geneva, we wouldn't have had it. So the instrument is very important because it's our means of investigation. Of course, you need to have the ideas. You don't build the Large Hadron Collider without looking for something. But you cannot look for it if you don't have it. So this kind of complicated mixtures can now be studied. And you don't throw it down the sink anymore. So first of all, it might be of interest for developing ways to get biologically active substances. It might be of interest for dynamic nanostructures, I will not talk about that, and can be interesting for dynamic materials. So first of all, we have to define a type of reversible reaction. The widest one and the most widely present in chemistry and in biology is the possibility to make imine bonds, to condense an NH2 with a carbonyl to make an imine. Now, there are not many carbonyls in biology, but the basic process using amines is present. There are many amines in biology. You can also make other types, like what we call acyl hydrozones. I don't want to get too much into those. They're very interesting because they have a peptide bond and a dynamic bond. But anyway, this formation of a CN double bond can be reversed, and this is a reversible reaction. Okay? So what can you do? Let's look first at the possibility to search for drugs, for biologically active substances. First of all, one approach is to make one key for the lock. That's what you would like to do. You would like to have a lock, to look at it, to make the key, and it works. That is designed drug formulation, designed drug discovery. But you have to know the shape of the lock. You have to isolate the protein, you have to get the crystal structure, you have to know how it looks. And then you have to design the molecule which fits, you have to make it, and then you have to try it. That is very interesting, because if it works, you understand, but it may take a long time. So the other approach, which was developed and is much used in many companies, almost all pharmaceutical companies, is what is called the combinatorial approach. They say, look, you guys who want to make the one key, maybe it's interesting, but it will take you a century to get it. We want to be quicker, so we make it 10,000 keys. We have fast robotics, high throughput screening, and we test 10,000 molecules per day. And if the 10,000 first ones don't work, we take another 10,000, another 10,000. You have libraries of molecules. You can buy those from different companies and so on. And okay, you try. But it's quite discouraging. If you try to understand molecular recognition, you count on chance. Okay, it's informative, but look at chance. So the question then came up, why don't we try to combine the power of combinatorics, the generation of diversity, with the power of information, molecular recognition? Can we do that? How can we combine it? How can we do a dynamic combinatorial chemistry and not a static? That can be done in the following way. You don't look for a single key. You don't look for 100,000 or 1 million keys. You have just components of different shapes, different structures. They have a functional group, the black dot, which can link them together. So you generate all the possible combinations of these components, which gives you a dynamic combinatorial library, because these things all the time exchange back and forth. So even if they exist in different quantities or in low quantity, or maybe not at all in given conditions, the fact that they can condense gives you access to all the possible combinations. Okay? Now, next step, what do? You take your telephone and you call up Monsieur Le Chatelier. Who knows who is Monsieur Le Chatelier? The law of mass action in thermodynamics. It says that if something binds, it will be favored. That's simply expressed. So you add the receptor, and now, so this is that combinatorics, and then you add information, recognition, and the receptor will pick up what fits best, obviously. The most stable entity is the one which fits best. So the log does the job. It assembles the best pieces to make the best substrate for that receptor. And that is in principle a way to search for biologically active substances for drug discovery by letting the log assemble the key, by letting the target assemble the key. Now that looks like panacea, but panaceas don't exist in science. There's nothing has a general solution, always problems. Here the problem is, it's a bit long, but okay. The problem is that, of course, if you have many constituents, then the concentration of each is very low. So you need a binding which is very high, otherwise you don't see anything. So that's not so good. But if you see something, then it's a very good binding. So for instance, if your concentration is micromolar, 10 to the minus 6, you need at least a binding constant of 10 to the 6 or 10 to the 7 to get an effect. So you have a very effective screening, but only when it's very good. Okay, let me just go here and give you an example just to show you that the principle works. Carbonic anhydrase is an enzyme which interconverts CO2, water, and carbonic acid. It has an active site which has a zinc and a hydrophobic wall. This simple compound is a very good inhibitor, 1 nanomolar, 10 to the minus 9. That's a very good inhibitor. And it binds by a sulfonamide, which binds to the zinc, and a benzylamine group, a benzyl group, which binds to the hydrophobic wall. So what can we do? We took a simple problem at the beginning, not too many things. 4 amines, 3 aldehydes. One of the amines was benzylamine. We knew the answer, of course. The other aldehyde was sulfonamide. So we know that that should bind to the zinc. That should be the other part. But then you mix and you get 12. 3 times 4 is 12. Among these 12, this one here is closest to this very good inhibitor. Of course, we know that an imine here is not an amide here. But if the main interactions are the sulfonamide with the zinc, the benzyl with the hydrophobic wall, then this is a minor perturbation. So, okay, we said, let's go for it. And indeed, I just show you here 2 peaks in the 12, which we can find by chromatography, HPLC. And you see that this one, which is the one he's looking for, is about the same percentage as this one, which has the same sulfonamide component, but another amine here component, another one. So what is the conclusion? In absence of carbonic anhydrase, you have all 12. Then you add carbonic anhydrase, and what you would like to see is that this increases a lot. And because it will be eating up this piece, this will decrease. That's exactly what happens. So by adding carbonic anhydrase, you amplify very strongly the one which binds best in agreement with the law of mass action. But it also indicates that it works, that indeed this dynamic library makes all the exchanges possible, and the best fitting is the one which is amplified. So, there are many others. I could have talked about acetylcholinesterase, about binding to lectins, about other things, neuraminidase. This has been developed by different labs, but this is for more detailed discussions. What about materials? Now, that's again a stupid idea. Do you want to make materials which break apart? If this is a material breaks apart, I mean, it doesn't stand out very long. Huh? It's not exactly what you want. Of course, there are some materials who do not want to break apart. But if they can, maybe there's a new property appearing, which static, so to say, materials which are static, which are stable, don't possess. First of all, when you make molecules which are dynamic covalent, have a reversible reaction, or use supramolecular materials which have non-covalent interaction, then you can try by applying these possibilities of dynamics on the molecular level or supramolecular level to polymers to make dynamic polymers, which we call dynamers, dynamic polymers. That means this is one of the components. That's another component. Classically, you would make, for instance, nylon, polyamide, by connecting in a covalent fashion one functional group with another one and make the polymer. But you can also connect this with that in a supramolecular way by interactions, or in a dynamic covalent way by having this connection reversible. This gives now rise, perhaps you can hope, to new properties, like self-healing materials. Materials which will heal because these bonds are reversible. So if you break a material, it will heal. It will stick together again. They will be perhaps responsive and adaptive. Let's have a look. First of all, let's have a look at the supramolecular part. The reason why I start with that is that it was something which, when we introduced this in 1990, that's quite some time ago, but at that time, polymer chemists didn't like it at all. Because for polymer chemistry, it's a chemistry where you make polymers, nylon, polyethylene, polyvinyl chloride, all these kind of molecules which are fantastically important in our society and which are made by making covalent bonds which are not reversible. So, okay, it didn't pass very well to this community at the beginning, but they said, look, what is this thing? This is glues, this is not very interesting, they don't stick together and all that. But let's see what one can do. We made, it was in 1990, this object, one of the components, which in the center has a tartaric acid unit. I haven't time to get into that, but it has two recognition groups, one at each end. The complement to that, this is a donor of hydrogen bonding, acceptor donor. The complement is another similar thing, but where the recognition group is the complement to that. ADA is complementary to DAD. You mix them, and you get a chain. But this is now not a covalent molecular chain, it is a supramolecular chain, where the cohesion comes from 3 hydrogen bonds linking the subunits, the components together. To indicate that in fact it is quite a big change, that is a solid. This is a solid. When you mix, you get a liquid crystal. This liquid crystal has a number of interesting properties. One of them is to form fibrils. And in fact, it's a step where you can sort of try to understand how you go from a chain to an association of the chains to get fibrils, and thicker as concentrations increase. So this was a long story, and it developed in this 23 years and so on. And many people have finally joined, and very good work has been done, for instance, by Bert Meyer in Eindhoven, and many groups around the world. And supramolecular chemistry has now, supramolecular polymer chemistry has now become a very well accepted and very active field. But then something interesting happened. 1990, 23 years later, October 2013, I got a message from a small company, and the CEO of that company, a small company which is located in Eindhoven and part in Zurich, told me, yes, we have used your supramolecular polymer concept for what? For making cardiovascular implants. What they did is that for children which have a severe congenital cardiac malformation, which need a heart reconstruction, they have made implants which they have made for material using the principles of supramolecular chemistry, linking by hydrogen bonds, which then can lead to implants which are biodegradable because they are not strongly bound, and which sort of, you have to choose the right one, provide an environment where the cells, the normal cells, can invade, and in children that works well because they are very active, there are many cells, they can invade the tissue. And this was then indeed used in 2013 by Monsieur Bokeria, Professor Bokeria, a very famous pediatric cardiac surgeon, at the Bakulev Center in Moscow, which is, I didn't know about, I learned about that, and is a very well-known hospital for surgery on children having cardiovascular malformation. And here is Dominika, the first one who was treated in October 2013, 23 years later. And she's jumping around now. And there were 5 more have been done, maybe more now. This is when I would make the slide. And you should also realize what that means. I visited this morning the technology center here. You see, one needs these things, but you don't know what is coming out of it. And of the basic research, you don't know when there's an application. In 23 years, first, of course, the basic science was developed. Okay, you need that. You need the knowledge. But then somebody has to realize that maybe it's interesting for biomaterials. Then somebody has to change the components until they have certain properties. Then you have to find a process to make these implants. Then you have to find a place in the world where they are ready to do the job, the surgery. And of course, all the way, you have to find the money to do it. So how can you predict that? Impossible. So I think in basic research, you have to contribute to knowledge. And then we need also these people who detect what can be done, who do it, and who are ready to spend their time doing it, developing the technology, finding the money, and bringing it to the end. And so we are all hand in hand, and we must realize that we are not alone, that we do the basic research, but the other people will be helping us, engineers, technicians, managers, capital investors. We need those guys too, otherwise we don't have the money. So let me now indicate what in terms of material science, of course this is material, but it's biomaterials. Let's look at some other, choose 2 other properties, no, 3 other properties. It's not too long, but it will, right? If you want to make dynamic polymers, you need reversible reactions between the monomers, polyamines, making connections with an amine with an aldehyde or poly-IC hydrozones. Let me just give this as an example of dynamic polymers which can be formed and broken. First of all, what about modifying mechanical properties of polymers? This is NH2, NH2, and siloxanes in the middle. It's one component. Aldehyde, aldehyde, siloxanes in the middle. You mix those two and you get a polymer which comes from the connection of this NH2 with this aldehyde. That's the polymer. Now this is a totally uninteresting polymer, a terrible one. It's very sloppy, very soft, very stretchy, very nothing interesting. Terrible mechanics. But then what you can do, you take this and you add to it what you might call hard monomers. This is much less flexible. This is still flexible, but much less than chains which contain dimethylsiloxane. You add it in increasing quantity. And now, because these things can react with these bonds here, they will be going into the chain. So if you add 25%, it already looks significantly better. Okay? 50% now, it's rigid enough to not to bend down anymore. 75%, it's still flexible, elastic, but it's stronger. So you can convert a film which is at the beginning very soft, very stretchy, mechanically totally useless, to something which is progressively getting harder and harder and harder and tougher. Now this film in itself is not interesting, but it just indicates that you can modify the mechanical properties of your polymers by progressive inclusion of something else. What about optical properties? You will see this would be also approved. Now this one is also already approved that it works, but you can visualize that also. Suppose you have 2 polymers formed from the components AB. A is an amine, B is a carbonyl. Then you use another polymer, A prime, B prime. A prime is another amine, and B prime is another carbonyl. You make the 2 films, okay? Now, okay, you choose them in the correct way, but let's admit that this one is colorless, not fluorescent. This one may have some color, maybe slightly yellow, not fluorescent or not much. Then you superimpose. Now, at the place where they're superimposed, because of this possibility of exchanging, two new combinations can form. A with B prime and B with A prime. Because you have four possible combinations, AB, A prime B, AB prime, and A prime B prime. At the beginning, you have only two. So this perhaps is colored. This perhaps is fluorescent. And if it's the case, you will see it. So I had Japanese co-workers. We had a collaboration with a Japanese company, Mitsui Chemicals, and we did the basic work. They tried to see the applications. So they made a nice picture. They like nice pictures. There's a cat with a head and the ears. This is the polymer AB. Then the mustache, the eyes, and the inside of the ears is A prime, B prime. So what do? You heat, color, fluorescence. Only where it's superimposed. So that, first of all, shows that it works, that you get exchange at the interface between the two polymers, and it, in principle, makes also possible to use it. For instance, if you have a heating laser, you can heat at the interface. We haven't tried it, but in principle, I'm sure this will work. Or you can make a millefeuille, a stack, and you can write in 3 dimensions. So you can have materials which are optically modifiable by these dynamic properties. Last thing, what about making self-healing films? For this, we looked for Diels-Alder reaction, which was reversible. I don't want to get into details. This Diels-Alder reaction between a fulvene and a cyanoethylene is very reversible at room temperature. But let's not get into too much into details. So you can, sorry, what is going on? Oh, yeah. So you can make a polymer where this connects with that, two components which have the termini, and then you can have connections which are fully reversible all the time. They open, close, open, close, open, close all the time at 25 degrees. You can make a film, you cut it in two, you superimpose, you press with your fingers, and you can pull. It sticks together again, simply because at the interface these bonds have reformed, have exchanged across the interface, and then it sticks together. So you can make self-healing materials. So this was more or less what I want to tell you. I will come to the conclusion. I had another section, but that's for another time, where one can talk about extending these dynamics to networks. And I saw yesterday a very nice example where one can apply these principles, making networks and making systems which dynamically will select what's going on. Maybe there are some interesting results there. And I want just to conclude by sort of summarizing that this constitutional dynamic chemistry consists in developing systems where continuously these things interchange. They exchange their components, they recompose, they recombine, they reorganize, they expel components, they can incorporate others, and therefore they undergo constitutional variation. So you introduce into chemistry a notion which is selection. And selection is something which is in principle biological, but it's much wider. Biology uses the selection principles. And maybe say something, if you have a biologist sitting here, Darwinian evolution is, we know that living organisms do that. But before life existed, there was a purely chemical evolution. Because you cannot jump from particles to a living organism. You have to have progressive steps by which you go to the threshold of life. And this is only chemical. There's no life there. So complexity has to get into the game by itself, by this self-organization. And I can even claim that Darwinian evolution is chemistry. Because what is it? Changing a nucleotide somewhere in a genome. It's another nucleotide, so it's again a change in a chemical constituent in the genome. So that allows adaptation, and then in terms of applications, one can think of making adaptive materials, adaptive technologies, and these kind of things. So we start with molecular chemistry. Without molecules, you have no supramolecular chemistry, but you can build supramolecular chemistry on molecular by design. You introduce the notion of dynamics, where these objects can be made dynamic, so they can now have this property which they didn't have earlier. Of course, supramolecular chemistry always had dynamicity. Molecular has to be intentionally introduced. Then you can operate selection and open the area, open the perspectives towards adaptive and evolutive chemistry. Evolutive is another step. Adaptive means that it can adapt. But then, of course, if you remove the effector, it may just redistribute. Evolutive means that you take advantage of the first step to have a second and a third. So it's an adaptation which is sequential, where the first step prepares the stage for the second one. This can be done also for the chemist here. If you have imine bonds, you reduce them to amines, then they stick. So one can think about this too. But let me just say that we are very much now working on this area, and it has been developed in more and more laboratories now, one being that of Jeremy Sanders in Cambridge, UK. And I think this, also the way you look at chemistry is somewhat different. You look at it as a system where the objects are not there once and forever. They can change with the solicitations, with stimulus, physical one, heat, pressure, electric field, and with chemical effectors, protons, or basicity, acidity, and all these kind of things. So I think there's a lot to do there. And also in the build-up of complex receptors, of detection. Maybe one can make self-selecting detectors where you have a mixture of components, and where if you want to detect something, the one which detects that will assemble. If you don't make the detector anymore, it makes it by itself. And there are already steps along that in different groups around the world. Okay, I hope you will contribute to it, and I was very happy to be here. I was as I said, small is beautiful. You have a very nice building, a very nice setup, and you have to show these people in Barcelona that. Okay. This being said, I have nothing against Barcelona.
So thank you very much, Professor Lehn, for this very stimulating talk. So we can open the floor for a couple of questions, maybe. So is there any question? No question. Yes, Pedro?
Maybe comment on your opinion of what the theory and computer simulations, what are the challenges for theory and in the present and future in this field?
I get that question quite often. You see, it happens
I'm sorry.
Of course, no, theory and computations can help a lot. Now, once you know the field, you can sort of by the nose try to find out what's going to happen. But it's true that you may make mistakes, and you can be helped by computing some of those structures. In fact, the ones I showed, even the ones giving the cylinder, for instance, things like that, they are rather simple things. And there's not much theory needed because they're rather rigid. But in more complicated objects, and especially I think one should try to develop a computational area where you explore also dynamics and what may happen. In fact, Martin Kaplus, you know him, he's in our institute 50% of his time. He developed a method where you can explore the surface of a protein and then see which group can attach at which position. And I would think that if you use that possibility with different combinations, you might be able, in a complex mixture, to explore. Of course, you have to do it maybe you can even do it by first representing a dynamic library by the computer, that means generating all the combinations. That's something which has not been done. It would be a very interesting field. You have a mixture, and you generate all the combinations. And the computer first calculates all the combinations, and then tries to fit them to a target. That's not been done. It's something which would be very interesting to do. But you know, you cannot do everything. You have to choose. And there are enough computational chemists around the world who, in principle, could do that.
Thank you.
Any other question? No? Okay. So if not, we will now close the session. You will come and sit here with me and with the Dean of the faculty. We'll close the session.
Okay.
So just a few words for closing the session. I just wanted to thank everyone that helped me and accompanied to put together all this program for this symposium, especially the help from the university, the Vice Rector of Research, the Faculty of Science with here the Dean of the faculty, also the Societat Catalana de Química, and also the Institute of Computational Science. The only thing it's missing here is the capital investors, so the next time we will invite some of them. So with this, I thank you, especially Professor Lehn and Professor Carulla and Maspok for coming, and I hope you enjoyed the supramolecular chemistry. So,
okay, as a Dean of the Faculty, I would like to thank you all very much for the lectures today. Supramolecular chemistry provides a bottom-up approach to nanoscale systems with applications ranging from biology to material science, and this is very important for the people studying in this faculty, that, as you know, there are biologists, chemists, environmental science, and biotechnology. Okay? I am sure that everyone will agree with me that the three lectures have been very interesting indeed, and it is a very special pleasure as well to have had the opportunity to hear Professor Jean-Marie Lehn, and a great privilege for us to be able to have a Nobel Prize winner here. As the Dean, on behalf of both the students and the staff, I would like to give my sincere thanks to you all, and especially to Dr. Xavier Rivas and the Research Institute for give the opportunity to the faculty to participate in this symposium. This, I think, is very important, that the students form part of the academic community, and this has been a good opportunity for them. Je tiens à remercier, my French is as bad as my English, but I will try to say something. Je tiens à remercier le professeur Lehn qui a accepté de participer à ce colloque. Pour nous, c'est un grand honneur pour profiter de sa maîtrise et vous inviter à revenir bientôt. À bientôt, professor Lehn.
Merci beaucoup.
Merci beaucoup. Bueno, gràcies a tots per venir i espero doncs, que us hagi agradat el simposi. Jo penso que ha sigut una gran oportunitat i em sap greu perquè, de fet, havíem enviat un correu a moltíssims estudiants que no hi ha hagut l'assistència que ens hauria agradat. Però bueno, els que haveu vingut i haveu disfrutat, espero que els hi expliqueu. Doncs moltes gràcies a tots.